Thank you so much for everyone who's joining. We're going to be getting started in just a minute. We'll just first let everyone get settled in before we begin. All right. So thank you so much for joining us this afternoon. Our discussion of stem cell technology for ALS and pediatric epilepsy featuring Dr. Evangelos Kiskinis. And a special thank you to all of you and the Feinberg School of Medicine alumni and friends of the Les Turner ALS Center who have been able to join us. My name is Jordan Sund, and I am the Director of Development at Feinberg School of Medicine at Northwestern University. It is my pleasure to welcome you and to introduce our speakers. A few housekeeping items before we get started. Please use the Q&A feature to submit your questions. At the end of the presentation, we will have plenty of time to answer some questions. This presentation is also being recorded, and we will send a link of the recording to everyone who is registered today. Our speaker today is Dr. Evangelos Kiskinis, Assistant Professor of Neurology, Neuromuscular Disease, and Physiology, Director of the Stem Cell Core Facility at Northwestern University, Feinberg School of Medicine, faculty member at the Les Turner ALS Center at Northwestern Medicine, and New York Stem Cell Foundation Robertson Investigator. Born in Greece, Dr. Kiskinis studied in London, Switzerland, and then at Harvard, and he came to Northwestern in 2015 to lead his own lab. His research is focused on addressing fundamental aspects of the biology of human neurons in the context of physiological conditions and in the context of disease. His lab has two clinical interests, motor neuron disease and epileptic channelopathies. Their hope is that their disease model systems will help identify points of effective and targeted therapeutic intervention. As you will soon hear, philanthropy plays an important role fueling the work of the Les Turner ALS Center. Along with our longstanding partner, the Les Turner ALS Foundation, we are seeking to raise $10 million in endowment support by the end of 2024. Before remarks from Dr. Kiskinis, it is my honor to introduce Dr. Robert Kalb, Director of the Les Turner ALS Center, and Joan and Paul Rubschlager Professor, who has agreed to join us to share a few updates on the importance of the center and its research and clinical entities. Dr. Kalb came to Northwestern from the University of Pennsylvania to be the first director of the Les Turner ALS Research and Patient Center at Northwestern Medicine, chief of the Division of Neuromuscular Medicine, and a professor of neurology. Dr. Kalb was born in New York City, received his MD from Cornell University College of Medicine, and completed a neurology residency at Yale University. His medical training was followed by postdoctoral laboratory research in neurosciences at Yale, where he was supported by a Clinical Investigator Development Award. Dr. Kalb has had continuous support from the NIH for the past 30 years. His lab studies the basic mechanisms underpinning ALS using genetically engineered mice, primary neuron culture, and yeast disease models. His work is focused on the fundamental molecular processes that go awry during disease. The Kalb lab is passionately committed to bending the arc of disease and finding a cure for ALS through innovative and collaborative research. And with that, I give you Dr. Robert Kalb.
Hello, I'm Robert Kalb. I am the director of the Les Turner ALS Center here at Northwestern. I just wanted to give you a brief overview and then, leave you the vast amount of time to hear what Evangelos has to say. But the center is interested in combining patient care, clinical research, and basic science research. So we have a large multidisciplinary ALS clinic. We see more than 600 ALS patients a year, diagnosing and managing the disease. In addition, we have a robust clinical research program with almost a half a dozen individual clinical trials. We're trying different interventions to try to slow the progression of disease. And the third leg of the program is the wet lab basic science research program. This is really where the answers are going to come, to understand the fundamental mechanisms of disease. And we think that finding the targets for therapeutic intervention are ultimately going to lead to very robust therapeutics for ALS. So there are half a dozen labs at Northwestern that study ALS using a variety of different model systems. Evangelos Kiskinis is I wouldn't even say he's a rising star anymore. He just is a star. He's going to tell us about his exciting work using human cells from patients and other models of diseases in a dish. Evangelos.
Well, great. Thank you, Jordan and Bob, for the kind introduction. And also thank you to everyone who's participating. I'm happy you joined in. I'm happy you're interested and you care. And I always welcome the opportunity to speak to you about the work that we do in my lab at Northwestern University. So as was mentioned, I established my lab. I moved to Chicago and established my lab here 5 years ago. And in my lab, which is in the Department of Neurology, we are using stem cell-based technologies to develop models of neurological diseases with the goal of facilitating the design of effective therapeutics. And again, as Jordan briefly mentioned, we have two major clinical interests. The first one is ALS, Lou Gehrig's disease, and that occupies most of our time and effort and resources. But we have also acquired an interest in developing models for pediatric forms of epilepsies over the last few years. And what I'd like to do over the next 20 minutes or so is tell you a little bit about both of these diseases, tell you about where the problems I believe are in figuring out the best way to therapeutically interfere with these diseases, and explain to you some of the approaches that we are taking in the fight against the diseases, by illustrating to you one or two specific examples of ongoing projects in the lab. So very briefly, my disclosures down there, which I need to show. So the first thing I want to do is give you a little bit of background on ALS. I know that a lot of you are probably familiar with this disease. It's a devastating adult onset neurodegenerative disease with currently no effective therapies. There's two drugs in the market that are prescribed to treat ALS patients, and while both of these are seemingly beneficial, their effects are very, very moderate. So the clinical presentation of the disease is manifested by the selective dysfunction and eventual degeneration or death of a very specialized part of the human body, which is referred to as the motor neuron. These are cells that exist within the central nervous system of every individual, and most of them are found within the brain and the spinal cord. And what these specific particular cells do is they communicate the actions of our brains to our muscles. And through this purpose, they allow us to control our movement. They control how we walk, how we talk, how we run, how we speak, how we breathe, and so on. So in patients that are diagnosed with ALS, these cells become progressively dysfunctional and eventually die, and that is presented clinically as a gradual inability to move different parts of one's body, which is relentless. It spreads all over the body and leads to eventual fatal paralysis. The age of onset of these individuals is about 45 years of age, and unfortunately, the average survival from diagnosis is between 2 to 5 years. So there's a desperate need to develop effective ways of slowing this disease down and reversing its course. Now, why do people get ALS? That's a complicated question, but there's been tremendous breakthroughs. A lot of these have come from Northwestern over the last 20 years in identifying specific genetic causes that can lead to ALS. Broadly speaking, the overwhelming majority of people that get ALS are what we refer to as sporadic ALS cases. What that means is that there's no family history for these individuals, and probably the cause is a combination of their genetic background, which interacts in ways that we're not entirely sure with their environment. In about 10 to 12% of cases, there's a clear familial history, meaning that there's multiple family members in every generation that get ALS. And the work that's been done around those families has been incredibly useful, because we've uncovered a number of genes that, when they have mutations, they definitely cause ALS. And what's intriguing about this disease from a genetics perspective is that there's a number of different genes that can cause ALS. In fact, there's now more than 20 different genes that we know of that can cause the disease. An outstanding question that we are really interested in engaging and addressing is it all one disease, or are there many different subtypes under the same clinical umbrella of ALS, this clinical presentation? And the way we're trying to address this disease is we're creating models that I'll tell you about in a minute of each one of these genetic subtypes, and we're trying to understand how similar or different they are to each other. Again, work over the last 10, 15 years has strongly suggested that although there are these different genetic causes of disease, there are common pathological features amongst the majority of ALS patients. The details here in this slide are not important, but what I wanted to highlight is that there's a particular protein, which is known as TDP43, that is known to form these aggregates, these accumulations of within this very specific neuronal subtypes that I was telling you about amongst the overwhelming majority of all ALS patients. So this suggests that there are common mechanistic features between different types of ALS, and it's really important to try and understand how people that have different mutations, different genetic backgrounds, have different environment, grow up and develop in different environments, get ALS. How do they get to this common feature? So the problem with studying and understanding the disease is that it's really hard to access the cells that are affected. As I described to you earlier, these very particular neuronal subtypes known as motor neurons exist within our brains and our spinal cords, and it's not easy to get access to them, particularly an individual that has ALS. So scientists have resolved to two different approaches to study the disease over the last 50 years or so. The first one is illustrated here on the left, which is using patient autopsy tissue. So this is post-mortem after a patient passes away. Scientists have been examining their central nervous system. This has been extremely informative in understanding some of the primary pathology that leads to the dysfunction and eventual death of these cells. In fact, this aggregation of TDP43 that I told you about in the previous slide was initially discovered in these types of approaches. The other approach that scientists have been utilizing is to create animal models of the disease. And there's been a range of different animal models that have been used, from using baker's yeast to worms, to fly models, to fish models, to mouse and rodent models. And the way these models are created, usually we take a mutant version of a gene that we know causes ALS, we insert it in these different model systems, and then we ask how these motor neurons, which exist within all these systems, become, which exist within most of the systems, become dysfunctional. Now, again, these approach has been incredibly informative and very useful. However, all these systems suffer from a fundamental flaw, which is that they're not humans. And in fact, only within the last couple of years, we've come to appreciate how the biology of this very central factor for all ALS patients, this TDP43 molecule, is dramatically different between human cells and a range of these different organisms. So to overcome these difficulties, we and others have proposed an alternative approach, which relies on our ability to generate what we refer to as patient-specific stem cells from any individual we want to study. This fundamental technology was initially discovered in 2006 and actually led to the Nobel Prize for the scientists that discovered it. But essentially, this cartoon illustrates what this technology allows us to do. We identify a patient we want to study. We isolate some sort of somatic cell from this patient. That can be a little piece of their skin or a few drops of their blood. And through a complex process of molecular reprogramming, we can turn this blood cell or skin cell into what we refer to as a patient-specific stem cell, which is unique to that particular individual. It carries that particular individual's unique genetic background. And it has two important characteristics that no other cell type in the human body has, which is that we have the ability to make limitless quantities of these cells in the lab. And we also have the ability to turn them into any cell type in the human body using a combination of chemicals or other small molecules, including the cell types that are affected in the particular diseases we want to study. In the case of ALS, this would be the spinal motor neuron or the cortical neuron, as I was telling you about. But in cases of the pediatric epilepsy, it's different brain neuronal subtypes. So once we have these cells in our hands in the lab, if we can make them for an individual that has ALS and an individual that does not have ALS, we can compare them side by side under the microscope or using an array of different approaches that we've established to try and understand what is wrong with this particular neuron, what is wrong with this particular neuron that belongs to this particular individual. And if we identify a mechanism that's dysfunctional, we can apply specific drugs to try and identify drugs that are able to slow down the disease process in that particular individual. As I briefly mentioned, an additional method that we utilize in the lab, which has become available over the last few years, is our ability to edit the genome of this particular individuals. And what that allows us to do is, in cases where we know of a particular gene that's mutated for an individual, we can correct that mutation using genetic tools and ask whether that fixes the problem that we're studying. So now that I've introduced the technology and the overall approach that we take, I want to provide you two specific examples of ongoing projects in the lab that are related to two fundamental questions. The first one is something that I touched upon earlier, which is, are these distinct genetic subtypes of ALS the same disease, or are they different diseases with the same clinical presentation? And this is a busy slide. The details are not important, but what I wanted to illustrate with this is that, first of all these genes have mutations that can cause ALS. And over the last few years, it has become apparent that these genes can be grouped into different categories based on what they're known to do within these neuronal cells. The fundamental question that still remains is whether there's any interaction, whether there's any mechanistic connection between all these different genetic subtypes. So, and this is fundamentally important for the following reason. This is a nice cartoon that illustrates the population of ALS individuals, and it uses color coding to denote the causes of ALS. So the overwhelming majority of patients, as I mentioned earlier, are denoted with the black color. And these are individuals that are of a sporadic nature, i.e. we do not know of a clear genetic cause. These color-coded individuals are all individuals that have ALS, but they have ALS because they have mutations in different genes. And a fundamental question is whether they are similar or not. If we know that they're similar, then we can use that knowledge to select people to go under the same clinical trial, or we can select the right drug for the right individual. So to begin to address this question, just as an example, we selected individuals that have mutations in one particular ALS gene, a gene called SOD1, which was actually discovered here at Northwestern about 25 years ago. And we made patient-specific stem cells using the approach that I illustrated before, and we made patient-specific neurons from these individuals. We then were able to study these in the lab, and through a complicated range of experiments, we were able to determine that these neurons exhibit a dysfunction in a protein known as VCP, which in itself can cause ALS when mutated. So going back to this schematic that I was showing you earlier, through this approach, we discovered that these individuals that have SOD1 ALS share some features with these individuals that have ALS caused by mutations in VCP. And this is the black box that I like to use as a metaphor of somebody has a mutation in a particular gene. This mutation causes some sort of dysfunction, which causes defects. And what we found in this particular case is that within the black box is dysfunction that is known to occur in other ALS patients. Now, this is important for therapeutic development because we now know that if we figure out ways to restore the dysfunction of this particular protein that is known to dysfunction in these individuals, we can apply them also to these individuals, and we'll have one drug that works for the broader population. Of course, as always, we try and understand whether what we discover for these genetic subtypes of ALS is relevant for sporadic disease. And because the technology allows us to make stem cells from any given individual, we've also made them from a number of sporadic patients to study those in parallel. So this is just one example. A second example is related to this other critical question, which is, can we better stratify patients for clinical trials? Can we select the right drug for an individual who has ALS? And I always like to use this slide because it sends a positive message. This is a schematic that shows the number of clinical trials for ALS sorted by trial start year. And I hope what you can appreciate is that there's been a resurgence in the number of clinical trials that are currently ongoing over the last few years. In fact, in 2020, we had the highest number of clinical trials. And the message here is that there's a lot of promising therapies that are in development that are currently being tested around the world for ALS patients. But one fundamental problem that we have is we often lump all of ALS patients within the same group, and we often ask them to participate in clinical trials. Again, a fundamental question is there a drug that might benefit one patient but not benefit another? So to address this, I'm going to present to you a case study. This was an ALS patient. We're going to refer to them as patient 025 for the purposes of this presentation, who came to us. This was a male individual with no family history, about 45 years old, very healthy background. Initially got ALS in the lower limb and had developed very slowly progressing ALS disease. So we decided to take on this individual and try and understand whether there's something we can do, whether there's something we can learn about this individual using our approach. So initially we did a bunch of genetic analysis, and we discovered that this individual has no known ALS genetic cause, which definitively puts them in the cluster of sporadic individuals. We made their stem cells, we made their neurons, and then we studied these neurons in the lab using a range of approaches. And again, this is just some experimental data. These are neurons from that particular individual, or these are neurons down here, or these are neurons derived from two unrelated healthy control individuals as a comparison. So one of the assays that we employed to try and understand what was wrong with this individual is we looked at the this central protein that I was telling you about called TDP43. And the particular is not important, but what I wanted to illustrate is that we discovered using our approach that this specific individual indeed had a metric which suggested that this protein becomes dysfunctional in neurons that belong to him and not in neurons derived from unrelated healthy control people. So with this metric in mind, we then looked at ongoing clinical trials and selected a range of small molecule therapeutics or drugs that are either in trials or are on the market. And we asked whether these drugs had the ability to rescue this particular disease-relevant change in this person's cells. And the details, again, are not important, but what I hope to illustrate here is that there were some drugs that had no effect in rescuing this individual's defect. There were other drugs that had an effect, and there were indeed other drugs that had a very strong effect in rescuing this individual's cells. So this is just a proof of principle of an approach that we can take. And if we had the resources to do more and more of these and examine more and more individuals with this approach, we would get to understand whether our model can indeed predict what drug would be best for each particular individual. So I'm sensitive of the time, so that's all I wanted to talk about in terms of ALS. And I'll now move on for the last few minutes to talk about pediatric epilepsy, and I believe we'll take questions at the end of the presentation. So very briefly, when I moved here to Northwestern, there was a strong clinical interest in pediatric epilepsy, and pediatric refers to child epilepsy. These are a very aggressive diseases that kids are diagnosed with soon after they're born. And they're characterized by very, very early onset seizures. And you know, these are devastating diseases which have profound cognitive, behavioral, and neurological deficits. So seizures is just one part of the disease. These children have problems, neurodevelopmental problems as they grow up. And again, this is another a case where there's inadequate treatment options. There's several anti-seizure medication that's out there available, but a fundamental problem that clinicians that treat these children have is that they don't know what drug would be effective for a particular case. And they often have to resort to a trial and error approach where they provide a drug to a particular child. Sometimes it works, sometimes it doesn't. If it doesn't, they have to take the child off that drug and use another one, and so on and so forth. And this can have devastating repercussions for the child as well as for the families of these kids. So we became interested in this, and there's a subgroup of these cases where we know where the genetics are very clearly defined, and we know that there are mutations in particular genes that regulate how neurons talk to each other, how neurons fire, that these individuals are known to have. So I'm just going to show you one example, and we've developed a program to analyze a range of these cases. But again, I'm going to tell you about a case study. This was a child which was diagnosed with a very, very early onset pediatric epilepsy. The kid had a mutation, a particular mutation in this specific gene. We recruited him through our collaborators at Lurie Children's Hospital, isolated a few drops of their blood. And with a method that I showed you before, we made their stem cells, shown here in red. And in this case, we did use genetic engineering tools to correct this particular mutation that causes this child's seizures. And we now have in the lab the patient stem cells with a mutation and the patient stem cells without this particular mutations. And we can now differentiate or turn these cells into, in this case, brain cells that are affected in this particular disease. In the ALS, we turn them into spinal cord cells. This is just some examples. These are how these neurons look in the dish, and this is just the genetic data. This is the only data slide that I want to show you, but I think it's incredibly exciting from my perspective. So in a process known as electroencephalography, what clinicians do when kids have seizures is they use this instrument which has electrodes that allows clinicians to monitor the electrical activity of their brain. And these kids that have this particular disease are characterized by this particular pattern in their electrical activity of the brain, which is known as the burst suppression pattern. This is actually the data from a particular patient that we're studying. So when we make our neurons from this particular individual using the approach that I told you, and we put them in similar instruments that allow us to measure their electrical activity, incredibly enough, we identify the same burst suppression pattern that we see in this individual's brains in their neurons that we grow in the culture dish. This is an incredibly useful platform for us because, again, it allows us to go back and ask whether we can restore a normal electrical pattern, just like the ones shown here or here, using small molecule therapeutics. And in this particular case, we collaborating with a pharmaceutical company to develop these so-called antisense oligo technology molecules that will allow us to target the particular mutation and restore the electrical activity first in the patient cells in the dish. And if we can do that, then we can see whether we can employ the same approach to do it in individual patients. So that's all I wanted to tell you about. Last but not least, I wanted to highlight the number of people that are involved in this work. So there's a number of people in my lab. The majority of my lab works on ALS disease, and a small proportion works in these pediatric epilepsies. We have a number of collaborators across different institutions, both within as well as outside the United States. And last and very importantly, I want to acknowledge the funding agencies and the patients that participate in these studies and allow us to generate their stem cells and perform this work. I'll specifically like to highlight the Les Turner ALS Foundation that has supported my lab since its inception, as well as the New York Stem Cell Foundation that has supported my lab for the last couple of years. And I think this is a critical slide because it illustrates that in order to do this type of work, we require significant resources, both in terms of personnel, in terms of collaboration, and in terms of funding resources. So thank you very much for your time, and I'll be happy to address any questions that might have come up.
Thank you so much, Dr. Kiskinis. So we have received a handful of questions. I was going to start with, did you use CRISPR to delete the defective gene in the PEDCSZ patient?
Right. Yeah, that's a very well-informed question. The answer is no. So we use CRISPR in the lab to perform genetic engineering experiments. And we do that both in the context of ALS as well as for epilepsy. In fact, we've corrected now in the lab in total, I think, 7 different ALS causal genes. as well as several different pediatric epilepsy genes. And this is a critical tool because it allows us to directly ask whether the genetic mutation, the genetic insult, is the cause of the defects that we're studying, that we're seeing in these cells. So absolutely, yes.
Thank you. And then a question came in asking what happened with your Brainstorm project?
I'm sorry, say that again?
They are asking a question about the Brainstorm project. I'm not sure if that was
right.
referenced during, yeah.
Right, so maybe Bob can be helpful here. I believe that refers to a clinical trial that's, I believe is currently ongoing throughout the country for ALS with the use of stem cells. I'd like to start by clarifying that the stem cells that are used in that particular trial are a different type of stem cells than the ones that I talked about today. And Bob, who's a clinician, can tell you more about where that trial stands.
Sure. Thanks, Evangelos. Yes, so there was a phase 3 clinical trial. Phase 3 means that we're looking for efficacy, and it compared individuals with ALS who received an infusion of their own stem cells to see whether those people had a more profound slowing of disease than those who did not get a stem cell infusion. And so the particulars were that the investigators removed bone marrow cells and then separated out a particular population called mesenchymal cells, and then treated them with a cocktail of factors to turn them into stem-like cells, and then infused them into the spinal fluid that surrounds the spinal cord. The hope was that these cells would take up residence and have beneficial, healthful effects. So as you can imagine, this was an incredibly complicated and expensive study to run, but it was a double-blind, placebo-controlled phase 3 study with 100 patients with the infusions and 100 without. At the present time, there has not been a publication on the results, but there was a group meeting a month ago, maybe 6 weeks ago, from the president of the company. And unfortunately, there were no disease-modifying effects seen in the individuals that got the infusion of the stem cells. And so without looking at the primary data, it's always hard to know whether there might be a subgroup of individuals that actually did respond. But at a first pass, using the pre-designed endpoints, there was no beneficial effect of the patient-specific infusion of stem cells. It was a big deal in the community, something we were very hopeful about, and unfortunately, as presently configured, it does not appear to be having a detectable effect.
Thank you, Dr. Cal. Where do you see the most promise in terms of the first corrective therapy for ALS?
Right, that's a great question. So I would say, my personal view on this is that there are obviously, as I showed in that slide, there's a number of ongoing clinical trials. My own personal view is that the most promising ones are the ones that are directly engaging with a genetic target. There's a new class of small molecules that I referred to towards the end of my presentation, known as antisense oligonucleotides. These are molecules that have the capability to engage with the mutant transcript or the mutant gene that causes the disease and delete it in individuals. And I'm very excited about those approaches because they're rationally designed, and they target something very specific, which we know is causal to the disease. And there's been very exciting and efficacious examples of utilization of antisense oligo technology for other neurological diseases, the most prominent one of which would be spinal muscular atrophy, with some very exciting therapeutic benefits for this individuals that suffer from disease. So currently, there's two relevant to ALS clinical trials for using those molecules. The first one is for SOD1 patients. I believe that is about to wrap up. That's a phase 3 clinical trial that will wrap up this summer. So the preliminary data that came out of the phase 2, seem promising, but we got to wait and see what will happen. And there's a second trial, which I believe is still in phase 1, if I'm not correct, for C9orf72 individuals with that specific antisense oligo technology. And obviously that's something that we're very excited about, because C9orf72 individuals represent the largest known genetic pool of ALS patients. So if we have something that works for them, that would be really impactful for the community. Bob, what are your thoughts on?
Yeah, no, I agree that I think the genetic forms of disease are the ones that will fall the first. And I also agree with you that I think antisense technology, ASO technology, is likely to be the effective in the intermediate future. There are lots of problems associated with antisense oligos. It requires monthly spinal taps, and there are also some technical factors like distribution. So if you you inject the antisense oligo in the lower spine, how does it get to the brain? I mean, there are technical factors here, but I think that I share your view that this is the most promising approach. The challenge is that the bad stuff that's happening is happening way before a person initially comes to see their physician, and typically way before the person gets to see a neurologist and is diagnosed with ALS. And I think that what you really want to do is catch this thing early, before the rippling secondary effects have taken over. And if you just use the analogy of a car, if the problem was the radiator, you want to fix the radiator quickly. But if you let the car continue to drive with a battered radiator, valves overheat, and whatever, there are dozens of other things that go wrong with the car. So the earlier we can diagnose and treat a patient, I think the more likely it will be to be effective.
Right. That's a very good point. I guess I also, as you were speaking, I also realized that there's we shouldn't only talk about the genetic subtypes of ALS. That there's a very promising antisense oligo approach that would be beneficial for sporadic disease, which targets a gene called ataxin-2, which is not in itself a genetic cause, but seems to be implicated in sporadic ALS disease. So there is. I think everybody's acutely aware that sporadic disease is the biggest problem. But I think that the key to solving sporadic disease will come through understanding what exactly goes wrong in genetic disease. I think my personal view is that I think the genetic subtypes are sort of highlighting to us the biology that we need to focus on, which when becomes dysfunctional is a problem for this particular subtypes, the motor neurons that degenerate in ALS. So the hope is that something that's relevant for these genetic subtypes would also be relevant for sporadic disease.
Clearly, I agree with you, because you and I have had this discussion multiple times. In fact, Evangelos and I wrote a grant together specifically to look at sporadic ALS and the model systems that he uses and the model systems that my lab uses as well. So, yeah, I mean, I think that it's a complex problem, and I agree with you as well that once we conquer the genetic forms of disease, it will inform us about sporadic disease. But I think the point you make is trenchant, that we really don't know how many diseases ALS is, and whether it's going to be biomarkers or stem cell technology or other ways to subclassify people into groups so that we can better target the therapies, I think will be an important step forward. I'd say right now it's not crisp how we're actually going to get there.
That leads us to, you spoke a lot about the patterns that you are beginning to see across certain genetic markers. And we received a number of questions around the application for other diseases. So things like any application to the central auditory processing disorders. Are there treatments with stem cells, specifically including RA and MS? Can you speak to how there's any applicability across the various spectrums of the disease?
Right. So I guess the approach that I described that we take in my own lab of creating these patient-specific models of disease by creating the patient-specific stem cells is one that has been applied across a range of neurodegenerative diseases, including Alzheimer's disease, Parkinson's disease, MS, as well as other rare diseases. So I certainly think that it is certainly true that the approach is broadly applicable, as long as we have the means to turn the stem cells into the particular subtypes that are relevant for that particular disease. And thanks to a lot of fantastic developmental biology that has been done over the last 50 years, we have the information to do that. So again, in terms of the therapeutics, the Antisense oligo technology that we discussed, again, has been broadly utilized across neurological diseases, including some forms of epilepsies, neuromuscular diseases that we talked about. And there are several promising clinical trials on that end as well.
Yeah, I'm just to chime in, I think that all the neurodegenerative diseases, the big ones like Alzheimer's disease, Parkinson's disease, ALS, so certainly Alzheimer's and Parkinson's disease are far more prevalent than ALS. But they also share features of selective neuronal vulnerability. A specific population of nerve cells is degenerating. And in addition, they share some other features when we look at the tissue under the microscope from an autopsy of clumps of misfolded proteins. It's as if the trash is not adequately taken care of. And so it may be that if we can get a rigorous understanding of how to manage the trash or how to manage energetics in one disease, it will have broader applicability. That's kind of the hope. And so it doesn't mean all neurologic diseases have that same feature of misfolded trash buildup, protein buildup, but that's a very common thing. And we think that's going to be important in a more broad sense.
Thank you. So switching back a little bit back specifically to ALS, someone asked, what are you doing to diagnose ALS earlier? And does the molecule mark defective cells before clinical symptoms appear?
Right. So great question. So in my own lab, as well as I think through the community, there are efforts to discover novel biomarkers of the disease. And this is another example of where these this patient-specific stem cell models can become useful, because it's a tool that allows us to see the early stages of a neuron that has mutations or has ALS. So the idea that we're exploring is we're trying to identify certain molecules that a disease cell secretes or produces before it actually becomes dysfunctional and starts degenerating. And the idea is that if we have something that looks robust in this model systems, we would then obviously go back and see if it's something that we see in pre-symptomatic patients. So I think one such piece of data that we were involved with previously was this differences in the excitability of individuals that have ALS. So work that has been done by a number of groups over the last few years has shown that pre-symptomatic ALS cases have excitability in the way that their brain cells, excitability differences in both in their brains and their peripheral nerves. And this is something that we're pretty excited about, in defining mechanistically in a project we have ongoing in the lab. But the key to this is discovering biomarkers, early biomarkers of the disease.
So this is a good example of how integrating what's going on in the basic labs and what's going on in the clinic is really absolutely fundamental. So, when Evangelist Labs or another lab across the country, but here at Northwestern most easily, identifies a potential handle as a as a biomarker, something we could look at in our patients, then this is a great way for the clinic and the basic science labs to be talking to each other so that, but at the present time, ALS is a clinical diagnosis. It's a diagnosis made by a neurologist by excluding other diagnoses and watching over time. But I think that the development of biomarkers not only is helpful diagnostically, but it can also be used to monitor disease progression. So if we have some objective measures that we can see that tell us this person is getting worse, above and beyond being weak, there's some other molecular feature, let's say something in their spinal fluid, that's something we can follow when we intervene with an antisense oligo or another medication or another intervention. So the more that the labs and the clinic are talking to each other, the better our patient care can be. And it can also inform what the basic science labs should be looking for and be thinking about. So it's a critical aspect of the integration in our centers, that both the taking care of the patients, the clinical research, and the conversations that are ongoing in the basic science research, we need to all be in the same room.
So I think we only have time for one more question. So apologies if we haven't been able to get to yours yet, if you submitted one. But what are your greatest aspirations in the ALS space for the stem cell models you're developing? And what do you need to be successful?
Right, that's a great question. So my aspiration is to develop a effective therapeutic for ALS within the next 10 years. I think the answer to the second question is pretty simple, and it is resources and collaboration. These are the two fundamental requirements that will allow us to get to that point. And I'm terribly excited by the fact that my lab is here at Northwestern, because we have the platform, the capability to execute on this plan based on both collaboration and resources and people that are interested and care. So.
No, thank you so much, Dr. Kiskinis. We really appreciate all that you're doing at Northwestern and on behalf of all of the patients who do have ALS. A special thank you to Dr. Kalb for joining us this afternoon as well. And a special thank you to our partners, the Les Turner ALS Foundation and Feinberg School of Medicine alumni who have joined us today. If you would like to learn more about Dr. Kiskinis' work or the Les Turner ALS Center at Northwestern, please do not hesitate to reach out. We would be happy to speak with you. All right,
Fantastic.
You all.
Thank you again for the opportunity. And if there are any outstanding questions, please feel free to reach out. Thank you again.
Thanks.